formed, there was a big dumping of CO2 off from that had been laid down earlier in the Carboniferous period. All the coal was burned off in these massive volcanic eruptions. So CO2 got dumped into the atmosphere. It got really hot. The oceans tried to absorb as much as they could, but they got to the point where they could no longer, the bryozoans back then, rather than corals, could no longer lay down calcium skeletons because things got too acidic. And once the oceans couldn't absorb it all, it got really hot. The average temperature at the equator about 250 million years ago was around 40 degrees Celsius. And that means on a regular basis, you were hitting 55 Celsius, which is not a temperature that vertebrates survive very well at. So that was the biggest extinction in the record that's out there. And what happened to the mammal lineage is we went from being the big dominant things back in the Permian to being these little things that for the next 150 million years were 180 million years were really less than 100 grams and hiding in holes while the age of the dinosaurs came around. And then we had this other event here. It's actually not as simple as a meteor smashing into the planet was definitely a big deal, but there was also a major volcanic eruption as India crashed into southern Asia, and there was a major volcanic dump. And by the time the meteor hit, there was about 23% less land mass because it was warm at the time. And so that was also a part of it. But that's when the mammals really radiated at that point. And so when we look at mammals, you know, the marsupials had already branched off by the time things had come into it. We'd already had the platypus and the echidna, and then the marsupials came off. There's 4 major orders of mammals, superorders of mammals. The Aphrotheria are like elephants and manatees and aardvarks and things like that. There's the Xenarthra, which are sloths, armadillos, and anteaters. And then there's 2 big specios groups. We're one of them. We're one of them over here. So this is the euarchonoglyres are easy to remember. It's primates and rabbits and rodents. And rodents are a huge part. If you just say bats and rodents, you've described most mammal species right there. That's more than half of the mammal species. And over here is the Labrasiotheria, which has the whales and ruminants, the horses and rhinos, the carnivores and pangolins, and most importantly, the bats, who are the most species group in there. And so I've got the 2 big groups represented on here. Now, when we look at how bats are related to each other, you can see, so meteor hit about 66 million years ago. Right after that, there's this massive radiation of diversifying species. And, you know, at first we thought that there were Megachiroptera and Microchiroptera, with the Pteropodidae being what we thought of as, but it turns out some of the other bats are actually more closely related to the pteropodids than that. So these are now the Yingchiroptera, and pteropodids are best known of the group. The rhinolophids are another group that I'll talk about, and these are the Yangchiroptera over here, where the things like Vespertilonids and Philostomatids sit. So the rhinolophids, I'm going to talk about first, but then that's going to be the last we're going to hear about them, are the horseshoe bats. They're old world leaf-nosed bats. There's only one genus, but it's got 106 species in it. And these guys are the hosts for the sarbecoviruses. So the things like the first SARS and MERS and probably SARS-CoV-2 are found in these guys. However, that having been said, these are really the only bats that are good hosts for them. Other experimental infections that have been done with SARS-CoV-2 and other bat species, they're not competent hosts. So I work with a lot of collections where I do worry about things getting COVID, like otters and primates and deer and things like that. But I don't worry about non-rhinolophid bats picking up COVID, which is nice at this point. It was definitely a concern at the beginning, you know, in 2020. But with some better data, we're seeing that and the rhinolophids are, I've never actually had my hands on one. They're not in North America. There are no captive rhinolophids in North America that I'm aware of. and they're native to Eurasia. So that's not as much of a, so basically what the rhinolophans mean is we don't get to play with them here, but it also means we don't have to deal with sarbecoviruses being endemic here. So a group I do play with a lot out of Luby are the pteropodids. They're the largest bat species. Some of them are known as the flying foxes. There are some small ones, like a little Cynopterus are not much bigger than a Vespertilionid. But the flying foxes are all old world species. There are 46 genera and 106 species. And they're mostly frugivores. There are some that go more for nectar than fruit, but in general, they're not really carnivores. And that provides some challenges from a diagnostic standpoint that we'll talk about in a little bit. The Phalastomidae are the New World leaf-nosed bats, and there are 55 genera and 174 species. This is where, so these are Artebius here. We see in North American, a number of collections have Artebius species, many of which are labeled Artebius jamaicensis, not all of which are necessarily, there's some diverse species in the genus Artebius, not all of whom were properly recognized when they came in. So we have a lot of mutt bats in captivity that are hybrids of species. But Artebius is a frugivorous bat who are in the Phalastomidids, or Carolia perspicillata is another pretty common one to see in captivity. But there's definitely some diverse habits amongst these. So Desmodus rotundus, the common vampire bat, is a Phalastomatid too. It's a sanguivore. Or Phalastomus cystatus is a pretty aggressive carnivore that will happily take some small vertebrates too. And so there's a lot of diversity to their lifestyles and how they approach it. And they tend to be a little larger than the Vespertilonids. The Vespertilonids are probably what most of the people who are working with native bats here in the US are working with. They're the simple nosed bats. They don't have big complicated faces that you see on some of the leaf nosed bats. There are 58 genera as of my last count when I went and looked on NCBI taxonomy browser, with over 300 species. They're all carnivores. Most of them are eating insects. Some other arthropods every now and then. So a couple of quick thoughts. One, anytime you're seeing a species that's presenting to you with a problem as a veterinarian and it's not a cat or a dog, husbandry is playing a role. Well, even when it's a cat or a dog, husbandry is playing a role with the animal presents with a problem. And so your thought process shouldn't be, is the husbandry okay? Because if it's presenting with a problem, it's not. So your thought process needs to be, how can I fix this husbandry? And rule number two, without fixing the husbandry, you're not going to fix the problem. So when we look at, and we were talking earlier, when I was a kid listening to, first learning about Darwin and evolution, I thought that survival of the fittest meant the smartest and the fastest and the strongest. And it turns out when you look at a genome, that's not at all what it means. What evolution has been time and time again is a series of selective sweeps of new pathogens coming into a population and taking out the susceptible ones. And the genes that are pushed the hardest to evolve most rapidly are generally immune-related genes. So what evolution really has been is who has survived the most recent plague. And when you look at it from that standpoint, you're probably in many ways better off not being the last one to get something. You're better off with greater mobility as a species. And so when you look at bats, bats are flighted. They are very capable of moving around to different places. Lots of them are colonial. They live in some of them live in huge colonies of millions. And so because of that, they're going to have things go through their populations more quickly, and they're going to evolve. And they've got an advantage that way. And so you'll often hear people talk about how bats are these super animals that get all these viruses and they're incredibly resistant to all of it. And the reason they can do that is not because they started out any better, but because all of the susceptible bats are long gone, and what we're seeing are the survivors. And that's what evolution has been, is a series of survivors of selective sweeps of pathogens coming through populations. Now, when we look at immune responses, we hear a lot about you have innate immunity where you just kind of, some things get recognized, but more importantly, we're looking towards specific immunity, where you recognize something that you have seen before. And there's two approaches to it. One that makes a lot more time on the popular press is humoral immunity. And so B cells make these antibodies, are often drawn as little Ys, and each one is actually, each B cells are actually recombining its own genes, and it picks a little V segment, a little D segment, a little J segment, it smushes them all together, and you have this variable portion at the top of the antibody that might recognize something. And at the bottom, there's an FC portion, which is the immune system handle. So B cells dump those things out, they go and latch onto things, and now there's an immune system handle attached to it. And that's great for finding things that are floating around outside of cells. But it's not the only way that interacts. Cellular immunity is about your T cells, and they have T cell receptors, and they undergo recombination very similar to how an antibody gene does that. And they pick a V segment, a J segment, splice them together, and each T cell is unique and signs things. But we're a T cell C stuff. Every cell in your body has these things called MHCs, and that says I'm self. And not only that, but it takes a little piece of proteins from inside it and puts it in a little groove and presents it on the outside. So MHC is saying, hey, I'm self, and here's what's hiding inside this cell. And your T cell receptor comes along, and if it sees that piece of protein it's looking for is a bad thing, it recognizes, it sets off this whole suicide pathway inside the cell. And so while looking at antibody titers is useful, it's not necessarily the big picture sort of thing. And for intracellular pathogens like viruses, cellular immunity is often a much more important part of the response. So when we're talking about infectious diseases and bats and concerns, the first thing that jumps to a lot of people's mind is rabies. And rabies is often taught like it's one virus. When you go over here and we look at lyssavirus, right? So here's the silver-haired bat rabies. Here's the Australian bat lyssavirus, which Australia is very careful to say that they don't have rabies. They just have Australian bat lyssavirus, which you can get from an animal with Australian bat lyssavirus when it bites you, and then you die of encephalitis. Duvenhage is a European lyssavirus. A lot of you probably learned about the R-OT when COVID first came out, which is how often a given infection spreads and infects somebody else. And it turns out that in order for these 5 little genes to go into an animal, track along the nerves, go to the brain, track along the salivary glands, go to the salivary glands and shed, at the same time you're doing behavioral changes, you actually have to be somewhat host adapted. So if a raccoon gets a skunk strain of rabies, it's probably going to die of rabies, but it's probably not going to be a very effective vector, and it's going to, on average, infect less than one other thing. So every human case of rabies that dies goes off to CDC and gets sequenced. And it turns out that a lot of diversity is present in a lot of bat species, but they're not all the same. When we look at, so in most of the world, it's going to be canine rabies. It's going to be the big killer. But here in North America, our canine population is much more vaccinated. And so that's not usually what people die from. In the United States, about 70% of the human deaths are from a strain that's associated with a silver-haired bat. Now, how much you guys have worked with silver-haired bats? They're very pretty little bats, but they're solitary tree nesting bats. And when you look at just about anybody's caseload, what they're seeing come in, the bats that come into contact with people are cave-dwelling bats like Aptesicus and Myotis and Tatarita and things like that like to come into people's houses in significant numbers. And those bats, when I was in Minnesota, we would see Aptesicus come in. We would probably get 100 of them a year. We would have 5 to 8 of them present with neurological signs. We would submit them. They would come back positive for rabies. It's pretty rare to have someone die from Aptesicus rabies. However, so my word of caution to you guys would be that if you're seeing silver-haired bats, you should treat that with more caution than you would others. Not that you should be cavalier and go getting chewed on by anybody, but a silver-haired bat is a higher risk for its strain of rabies actually causing significant disease in you. That having been said, most of the remainder of the about 20% of the human deaths are from free tails, from Tatarita. So that's also a concern, but that's a cave dwelling bat that has a much higher contact rate with people. So here, this is Anne, and you can see Anne has stuff growing on her junk. And this was a problem. She first started having proliferative vaginal growths that were kind of cauliflower-like, and we tried debriding, but eventually it got to the point where it was obstructing your urination. You know, we diagnosed that this was consistent with papillomavirus-like things. Here's a closer look at her at a different time period. And we would treat it with cryotherapy, and we were trying to induce an immune response to it. But eventually this ended up killing her. And what Anne actually had causing this, it went, full squamous cell sarcoma. What Anne actually had, this is HPV-16, and this is a major cause of human cervical cancer. When you look at its closest relatives in the alpha papillomaviruses, all the close relatives are in primates, and then closest non-primate papillomavirus to it is this thing that killed Anne, and with a very similar pathology to that. And that paper is about to be submitted. Some other bat problems you might run into. There is a genus of coccidia called Nephrisos breptisicae, which is a big problem in the ptiscus. It infects their kidneys. It's from a group. When we look at the coccidia, you often think of things like Eimeriae and Isospora being intestinal sorts of stuff, but and that's the Eimeridae. But when we get to the Sarcocystidae over here, there's some things that are a little scary. You may be familiar with Toxoplasma from cats, or Sarcocystis, Sarcocystis neurona in horses and things like that. So they tend to be more likely to go to other tissues outside of the GI tract and cause some serious issues. So this is something to watch out for in them. And some of these are sporulated, but urine is actually where you're going to see them shedding coccidia, and that's definitely something to be aware of. So immune systems in bats have been finally honed by evolution and their group lifestyles and high degree of mingling with each other in large numbers. And as a result of that, they have immune systems that have been heavily selected to jump on things. One of the challenges we run into in humans, is that we know that it's not normal for us to be running around with our immune systems having nothing to do. It's everybody's grandma used to have, or grandpa used to have pinworms. It was a normal thing to have as a kid, when they were around. And now no one gets out and plays in the dirt, and the rate of human parasitism has gone down really low. Now, that's a good thing in the case of things like ascarids, but in the case of pinworms, which just gave you an itchy butt, that just means your immune system gets bored. And so when you look at humans and all of the kids who go anaphylactic with peanut allergies and all that sort of stuff, that's very much a first world problem. And it doesn't happen in third world countries until you deworm all those people. So if I had a way to get pinworms into the Tropus collection at Libby, I suspect we would see a lot less allergies. But you can see this is Mindy here. You can see her wrist is all ulcerated. I got a closer picture coming up. This is all ulcerated. This eosinophilic sort of response is kind of classic for allergies. I think this is Crow over here, and they get these nasty ulcerated lesions. We put them on a lot of antihistamines, Zyrtec, and various. There's a lot of individual variation as far as what works best for one animal. We end up trying several different antihistamines to see what works best for an individual bat. Another thing we have been doing is to do intradermal skin testing, try and figure out what the allergens are. So we generally go over the back and specifically inject different potential allergens to see what the causes are. And we've been using antigen therapy to try and induce tolerance in them. It's detailed in this paper. It would be wrong to talk about infectious diseases in bats without talking about white nose syndrome. So pseudogymnascus destructans is the cause of white nose syndrome, and it's been a significant problem in bats. But it's a problem in hibernating bats, which for Florida is less of a concern. Honestly, bats don't down here don't hibernate so long that they actually run into problems with this, because what ends up happening is it ends up irritating them, and they become active during hibernation and burn off their calories and die. So that's a significant problem, and definitely something to be aware of, and something you don't want to be spreading in a facility. But it's actually something I don't have a lot of direct clinical experience with. Now, if you're successful and take great care of your animals, eventually you're going to end up with old animals. This Rosettus here is you can see he's kind of hunched over. He's hanging on by his thumb. We call this cradling. He's hanging on with his thumbs as well as his feet. He looks like he's pretty sore and arthritic. So we end up in a lot of these older, and you would think too that if you hung upside down for a living, you wouldn't get spondylosis and stuff like that, but it turns out that's not correct. We see an awful lot of spondylosis in a lot of the older bats too. And that's just part of being successful with keeping young animals until they turn into geriatric animals. We are learning more and more about how to work with them and improve this. We go often very multimodal with our therapies. We have a lot of bats on NSAIDs. Bats tend to be fairly NSAID tolerant. I should qualify that. Pteropods tend to be very NSAID tolerant. We're lacking data for an awful lot of species though. We do a lot of tramadol in a lot of the older bats, and we do a lot of gabapentin in a lot of the older bats. And with bats that are behaviorally appropriate, we have, so this was Curry. She was an old girl who had some spondylosis, and she was getting laser therapy for inflammation in her joints, and really seemed to respond to that very well. She was also getting electroacupuncture once a week, and she would just kind of come in and get treated. And there was a very marked improvement in her mobility following treatment every week. Some other issues we see. So this is a Philostomus hystatus. It's one of the carnivorous philostomatids. They get dental disease is very species dependent. Philostomus hystatus have terrible teeth, whereas, for example, in Pteropus, almost all the dental disease they get staining, which isn't really a problem. They don't really get plaque in very often at all. And you see trauma. So that's the sort of thing, and just wear and attrition in old bats. It's not the same sort of thing. Whereas Philostomus get really horrendous dental disease. And amongst the vespertilonids, the worst of the bunch is Aptesicus. I mean, Aptesicus just get really bad dental disease. And it probably has a lot to do with our diet. We're unable to really replicate. I believe they're primarily beetle eaters in the wild. And we feed them mealworms and things like that, but it's really hard to replicate an insectivore's true diet in the wild. And we're working on continuing to improve this, but you can see these teeth are just completely caked in stuff. You can get in there, and we use an ultrasonic scaler. You have to obviously set it a little lower so you're not vibrating things too much. We can get in there with an ultrasonic scaler and clean those teeth up. So those are significantly improved after regular dentals. But unfortunately, a large part of this is probably nutritional and normal oral flora that we're just not meeting what they've evolved to do, and we need to figure out how to do that better. Nails, it's not uncommon to see in these guys some nail issues. Some some of them will lose their sheaths. We will see broken toes and things like that when they get caught on things. Another not uncommon thing to see is a deep digital flexor tear. So when they have basically for the third phalanx that coming out here, there is only one flexor, the deep digital flexor, and if that gets torn, then they can't flex the end of that nail. And if you hang by your toenails for a living, that's really important. Another issue we have seen in a small number of bats, but it's enough that I'm very concerned about it. So I don't know if anybody here does horses, but horses will get laminitis, and the bone will come away from the outlying hoof, and we've had a small number of bats do exactly that, where you can see this bone is rotating right out through the nail. And they seem to get really inflamed, and sometimes they'll slough the whole nail, and they've got nothing left to hang by. It's been a couple of bats that we've seen this in. It's been a significant concern seeing a laminitis like this, but we haven't yet had a. Seems like once they really get this, it's really hard for them to come back from it. Other things we'll see in bats will include dependent edema. So in people with heart failure or something like that, you see their legs stock up and things like that. That's because that's the part that is downward. In a bat, it's generally the head that's downward. So this is a bat with pretty severe edema. There's a few different reasons that may be happening. It's generally involved protein loss somewhere is a common reason for it. So renal failure bats will sometimes do this, or bats with severe enteritis. But the most common reason we're seeing this is cardiac insufficiency, which is like in humans. And so that's certainly a big concern. They're just, you feel the head is just all edema and poofy with fluid. And this is something that generally presents in older bats. We're currently working on, that's not a movie. Getting normal echocardiographic measurements. We're now recognizing more and more that we are seeing some hypertension in bats. And we have been treating some of the bats with hypertension and have had some positive responses. One of the challenges that we run into, so here's a bat being echoed by one of our cardiologists. This is a bat being prepared for anesthesia, is being intubated. For this, we monitor them. Typically, we will have a Doppler looking at blood flow. We will be monitoring end tidal carbon dioxide with a capnograph, and trying to maintain normal blood gases. One of the things we have done with some of these bats who are showing this hypertensive syndrome like that is to measure blood pressure. Now, unfortunately, there's not a good place to put a cuff on a bat, so we have to directly catheterize arteries and get direct arterial pressures in them. It works pretty well. It is much easier to do in bigger bats. We have not done it in vespertilinids. Other things you can do, we see fractures that go into these guys, and you definitely can do the smallest of bats. You can repair a fracture. So that's a pretty straightforward thing to do. We wrote some of it up here. It is certainly possible to go. We see some neurologic injuries. Sometimes a bat will injure its brachial plexus traumatically. We can check and see that we have appropriate nerve function going on there. The other things that are important to recognize about bats, coagulation profiles are very different in bats. They're different in birds too, things that fly and things that dive. So they're different in whales, they're different in pinnipeds. There's kind of two pathways for coagulation. One is the extrinsic coagulation pathway, which is dependent on damaged endothelial blood vessels, and the intrinsic coagulation pathway, which is dependent on turbulence. And if you're something that flies or dives or has weird pressure things going on, the sort of blood pressures you need to fly might set off an intrinsic coagulation pathway. So we know that Pteropus have a clotting factor 11. Their gene is truncated. It doesn't completely go. So it's more challenging to look at clotting profiles in bats because of this. Another factor that's weird when we're looking at chemistry profiles is that one of the things we often look at in cats or dogs or humans to say, how are the kidneys working, is how well is this thing dumping out urea? Is it building up, or are its kidneys able to eliminate that? And in a bat, they have a fairly low purine diet, and being a frugivore like that makes it hard to build up levels. So even in bats with severe renal disease, we're not going to see the sort of elevations in BUN and creatinine that we might see in a carnivore, and that makes diagnosing renal disease somewhat more challenging. I just threw that in here because this in here towards the end, because it's kind of a fun picture. This is a bat that we just worked on recently. We're looking at a barium swallow, fluoroscopically. This bat has been having some issues swallowing food and seems to get kind of plugged up. And you can see as you watch, she will form a food bolus, but it isn't all necessarily. You can still see there's a big chunk that just kind of stuck there. And so she has some esophageal motility issues. She's an old girl, she's 31, but compared to even some similar age bats, it was very different. So we are working that up as we move forward. And this was a spectacled flying fox. And I think that brings me to the point where I should probably stop talking. Here we go. Let's see. All right. How was that?
Oh, thanks, Jim. We got time for a couple questions, I think. First question, what's the difference between old world and new world bats?
Well, it's not necessarily, there's not one I mean, where they live is the short answer to that. They're not all, you can see how that rapid radiation right after the KT extinction, they really have been several different groups that have been evolving independently for quite some time. And there are, some old world speed, like there's vespertilonids are all over the world, except for Antarctica. And they have kind of spread all over, whereas, pteropods are exclusively not in the Americas. So really it's just where they've evolved, because at this point they have mixed significantly in different parts.
Okay, thank you. Next question. Do you recommend the use of neutrophil or lymphocyte ratio determine the effects of either extra or intracellular pathogens?
Oh, unfortunately it's not that simple. Because I mean the main cell recognizing things in cellular immunity is a T lymphocyte. The main cell involved in producing antibodies is a B lymphocyte. And so the sort of overall type of response you're going to have, but it doesn't really tell you whether there's a cell division. And one of the challenges is it is fairly cheap to go and do a antibody titer. We did a study a few years ago too, also showing that there's a protein that grabs onto the FC portion of antibodies to measure them and do things like ELISAs and things like that. And we showed that this staph protein A works really well at least in a few different pteropods to look for antibody responses. But if you're looking for cellular immune responses, those are not easy to do and fairly expensive and require things like live lymphocyte culture. And even in human medicine, where it would only be done under like a referral immune clinic and be thousands of dollars to do. So unfortunately, most of the time when we're looking at immune responses, we're looking at antibody responses and missing part of the picture. And it's important to recognize when you're seeing part of the picture that you're seeing part of the picture and not the whole thing.
Okay, thank you, Jim.